Hello and welcome everybody to my channel ominibioentrance, and today we will be discussing CSIR PYQs for the year 2019. I will be discussing the applied biology and methodology section. If you are new to my channel, please do like, share and subscribe. So coming to the year June 2019. A multimeric protein when run on an SDS gel showed two bands at 20 kDa and 40 kDa. However, when the protein was run on a native gel, it showed a single band at 120 kDa. The native form of the protein would be. So here, a protein was run on SDS gel, it showed two bands 20 kDa and 40 kDa, but the native protein was found to be 120 kDa. So what is the native form of the protein? Whether it is a homotrimer, heterotetramer, heterodimer or heterotrimer. So here it is 20 plus 40, that is 60 kDa. You would already think that it is a heterodimer, but then the native gel it showed 120 kDa. So there are two 60 kDa bands involved. That is 2 from 20 kDa and 2 from 40 kDa. So it is a heterotetramer. Answer is option 2. Then next one. Orientation of a cloned DNA fragment in a plasmid vector can be checked by. So it can be checked by PCR using combination of one gene specific primer and one vector specific primer. So this is the diagram for that. So this is the gene of interest. So here this is the vector. So vector, this region will be your reverse primer and here it will be your forward primer. So it will get amplified. Now, if it is in the reverse orientation, this will be the forward primer and here it will be the reverse primer and this region will get amplified. So, PCR using a combination of one gene specific primer and one vector specific primer is the answer. Then next one. Based upon phenotypic observation, it was concluded that an unknown gene responsible for an agronomically important trait is present in a particular plant. In order to identify the gene, a researcher proposes to use the following strategies. PCR amplification of the gene. Now if he uses PCR amplification, then no PCR can be conducted because the gene is unknown, so we cannot develop any primers. So PCR amplification is not possible. Map-based cloning of the gene. So it can be used only when we know where the particular gene is located. So that also cannot be used. Subtractive DNA hybridization. That is correct. So genome sequencing also cannot be used. And develop molecular markers linked to the trait also cannot be used. So the answer here is option 3, C only. Then next one. A T0 transgenic plant contains two unlinked copies of the tDNA, of which one is functional and the other is silenced. Segregation of the transgenic to non-transgenic phenotype would occur in a dash ratio in progeny obtained by backcrossing and in a dash ratio in F1 progeny obtained by self-pollination. So, segregation of the transgenic to non-transgenic phenotype would occur in a dash ratio in the progeny obtained by backcrossing. Backcrossing will give you 1:1 ratio. And self-pollination will give you 3:1 ratio. So the answer here is option 2, that is 1:1 and 3:1. Then next one. Following statements were given regarding factors influencing variation in expression levels of transgene in transgenic plants. Which of the following options represent a combination of statements that would not lead to variation in transgene expression? levels in the transgenic plant generated using the same tDNA binary vector. So we will see the statements first. Difference in restriction enzyme sites within the tDNA. It does not make variation in the expression level. Difference in copy number of the transgene. That is correct. Where there is more copy number, there will be difference in the variation also. Variation in expression levels of the transgene. Then variation in site of integration of the tDNA within the plant genome. Whether tDNA is integrated into the heterochromatin or euchromatin, that will be a deciding factor. So C is also correct. Presence of multiple promoters within the tDNA region. There are no multiple promoters in the tDNA region. So option A and D is the answer. So the answer here is option 4, A and D only. Then next one. Run-off transcription assays were performed to establish the specificity of 3 novel sigma factors for their promoters. Results of the experiments are shown below. Following inferences were made from these results. So this is the experiment. We can see from the data that sigma A, it bind to promoter P2. Sigma B bind to promoter P1. Sigma A plus B bind to promoter P1 only. It may be because sigma B prevents sigma A from binding to promoter A2. Sigma C can bind to both the promoters P1 and P2. So we will read the inferences now. Sigma A initiate transcription from P2 and sigma B from P1. That is correct. A is correct. Then sigma C can initiate transcription from both the promoters. That is also correct. Sigma B prevents initiation of transcription from P2. That is correct. That is why sigma A plus B, sigma A cannot bind to P2. Then sigma A initiate transcription from P1. It is not P1, it is P2. So A, B, C only are correct answers. Answer is option 1. Then next one. Agrobac next year, that is December 2019, shift 1. Not next year, next month. Okay, shift 1. So first question. Agrobacterian tumefaciens is frequently used as a vector to create transgenic plants. Under laboratory conditions, agrobacterium mediated plant transformation does not require host plant genes that are required, bacterial type 4 secretion system that is also required, Vir genes are required, but opine catabolizing genes are not required. So answer is option 4. Then next one. The tDNA region of a TI plasmid of agrobacterium tumefaciens harbors 2 genes X and Y. Mutation of gene X produces rooty tumor, while mutation of gene Y produces shoots in the tumor. Based on the above information, which one of the following statement is correct? So if there is a mutation in gene X, it produces rooty tumor. That means X should be a cytokinin. When cytokinin gene got mutated, it resulted in the production of roots. And rooty tumor. And a mutation in gene Y produces shoots in the tumor. So gene Y has to be an auxin. So it results in shoots in the tumor. Based on the above information, which of the following statement is correct? So gene X encodes cytokinin and gene Y encodes auxin. That is correct. Answer is option 2. Then next one. Which of the following statements related to molecular cloning procedures is incorrect? 5' overhangs of restricted DNA fragments can be blunt-ended by clinopolymerase but not by DNase 1. That is correct. A DNA fragment obtained as an XHO fragment can be ligated at the Salvan site in a vector. This they had asked as a question of part C in one of the years. I have I have explained it over there. You can refer to that video. Then next one. To prevent self-ligation of a vector digested with KPN1, alkaline phosphatase enzyme is used to remove 3' phosphate groups from the ends of the fragment. That is not correct. So that is your answer. Alpha complementation. Blue-white screening may produce blue-colored recombinant colonies in case of translational fusion with the beta-galactosidase gene. The beta-galactosidase gene is not disrupted, so that option is also correct. Then next one. Given below are schematic representation of the tDNA region of 4 constructs that are to be used for agrobacterium mediated transformation to silence an endogenous plant gene represented as XYFP that is expressed constitutively in the plant. So you want to silence XYFP. So which construct you will be using? M is selectable marker, LB is left border and RB is right border. Which of the 4 constructs depicted above could be used to silence the target gene XYFP? So if you see the first case, there is XYFP here, there is XY here, that cannot silence, so that construct is not correct. In the second case, if you see XY is here, FP is here, intron is over in the middle, that also cannot be used for silencing. But if you see option C, it is PFYX, it is an antisense construct, it is in the XYFP gene in antisense construct. So C can be used for silencing. Here also, D also, if you see XYFP and PFYX, they can form short hairpin RNA will be formed. Then miRNAs will be formed and the result is silencing. So C and D can be used for silencing. So the answer here is option 4, C and D only. The next one. The following statements are made regarding developing a transgenic mice. Choose the combination of statements arranged in the correct sequence for developing transgenic mouse. So I will be reading out only the answer here. So first one will be the female mouse of specific strain is superovulated, oocytes are collected and allowed to fertilize in vitro. Then comes C. The desired gene is preferably micro-injected in male pronucleus after sperm entry in oocyte. Then E. Blastocyst stage embryos are transferred to the uterus of hormonally prepared mother. So answer here is GCE. Then next one. Given below are 2 sets of terms related to various methods used in biological science. So race is for determining the ends of the mRNA. A2. Southwestern blotting. Southwestern blotting is for DNA protein interactions. B. Recursive PCR. Recursive PCR is for construction of synthetic DNA and Tachman is FAM. So the answer here is option 2, A3, B1, C4, D2. Then next one. In an experiment, a 1 kb fragment with a single BAMH1 site as shown in figure A, this figure, is used to clone in the SMA1 site of the cloning vector of 3 kb length. This is the cloning vector, this is the promoter, this is the SMA1 site. None of the other enzymes of the multiple cloning sites are present in the fragment to be cloned. Based on the information given above, a series of digestions were set up for potential clones and the fragment profiles are given below. Which one of the above digestion profile confirms successful cloning of the fragment in the vector in an orientation wherein the 5' end of the cloned fragment is towards P? That is, it is in the sense orientation. This 5 prime end is towards P, means in this direction, 5 prime to 3 prime. So small one, you take and put the whole copy over here, and this is it. This is the diagram for the construct. I have not drawn it circular. Okay, so I am sorry about that, but it is a circular vector. So this is point 2, this is point 8. You have got a BAM H1 site in the insert and a BAM H1 site in the vector also. Now first option, option A. BAM H1 digestion, 200 base pair and 3.8 kb. If you digest with BAM H1, you will get a 0.8 kb fragment and then 3.2 fragment. So option A is not correct. Then next one, BAM H1, 800 base pair and 3.2 kb. That is correct. Hint 3 eco R1. Hint 3 and eco R1, if you digest, you will get a 1 kb and a 3 kb band. That is correct. But if this is in the inverted orientation, still then by hint 3 eco R1 digestion, you will get 1 kb and 3 kb base pair. They have already told in the question where 5 prime end of the cloned fragment is towards P. So if you use hint 3 and eco R1, and if the insert is in the reverse orientation, then you cannot distinguish between the reverse orientation and the right orientation after digestion with hint 3 and BAM H1. So hint 3 and eco R1. So that is not correct. XHO BAM H1. So that 200 base pair, XHO 200 base pair, BAM H1 800 base pair and then 3 kb. No, so that is not the correct answer. So the answer here is option 2, that is B only.